Hello, everyone. My name is Jules Nde. I am a fourth-year PhD student working with Professor Margaret Chen at the University of Washington. Today, I want to talk about protein-protein interactions. Specifically, I'm focusing on this protein called kinase 2. So calcium calmodulin-dependent protein kinase 2, kinase 2, is a multifunction protein. It's a multifunction protein which is involved in multiple cellular processes, including cell cycle regulation. Including cell cycle regulation, calcium regulation, and memory. So cell cycle regulation and the calcium regulation. So kinase 2 is activated upon binding to this protein called the calmodulin. And many experimental and theoretical results have shown that as the calcium binds to the calmodulin on 2, the affinity of the calmodulin is increased. So the affinity of the calmodulin for the kinase 2 is increased. And in turn, as the kinase 2 binds to the calmodulin, the affinity of the calcium for the calmodulin is also increased. But however, the basic mechanism underlying this complex process is still lacking. So, and also due to the large size of this kinase 2, the calmodulin-binding domain of kinase 2 is often used to study its binding properties. In this work, we are interested in studying how this kinase 2 interact with the calmodulin and how it will actually affects the calmodulin properties the calcium properties of the calmodulin. So we are doing so by using two approaches: the experiment and the molecular dynamic simulations. As you can see here on this one is not. But as you can see here, you can see that the graph of the calcium release from the calmodulin shows that as a function of time shows that in the presence of the calcium with no mutation. So here we are using three system, the system with three peptide system with no mutation or wild type, a single mutation and triple mutation here. So you can see that in the presence of a system with no mutation, the calcium affinity for the calmodulin is highly increased as compared to that of the free cam. So on the other hand, you can see that the system with triple mutation shows a high decrease in calcium affinity for the calmodulin relative to the wild type, as you can see here. So this highlights the importance of these charge residues RRK in the binding process of kinase 2 with the CAM. Additionally, you can see here on the right side that as the peptide with triple mutation bind to CAM, the free energy profile of the system is highly increased. So it's the shown here in blue dots is highly increased as compared to the wild type system. And this is confirmed by the 2D profile energy, where you can see that in the presence of triple mutant, the stability of the system is highly confined as compared to that of the wild type system. And we also conducted the residue room mean square fluctuation, and one can see that in the presence of the system with triple mutant, so we observe high fluctuations in a C domain of calmodulin, as you can see here in the C domain of CAM, as compared to that of the wild type, and specifically these fluctuations happen in the calcium binding site in the C domain. So this shows that so this can help to explain why in the presence of triple mutant we observe very high the calcium very high the calcium release rate from CAM. So as a conclusion for this work, we can say that as a CAM kinase 2 binds to calmodulin, the calcium affinity for CAM kinase 2 increased considerably. And also these specific residues called RRK, so which are the charge residues, actually contribute to the high calcium affinity for CAM through the electrostatic interactions with these residues and the C domain of calmodulin. I thank you for your attention.
Thank you, Jules.
Thank you.
And our last speaker, Sam Powell.
There you go. How should be working? Okay. Hi, I'm Samantha, or Sam. I'm a postdoc here at PNNL working with James Evans. So today I'm going to be talking about microED, or microelectron diffraction. So microED is a cryo-EM-based technique that we use for atomic structure determination from three-dimensional crystals. So here on this top left, these darker spots here are all three-dimensional crystals placed on an electron microscope or an EM grid. Actually going to back up real quick. There. Okay, so now this video is playing. So this is a tilt series here collected as the electron beam, as the stage rotates below an electron beam, you're going to collect these different diffraction patterns. So you have a series of images or a movie of diffraction patterns. So with these diffraction patterns, this is where we then end up determining a three-dimensional structure. So microED is a very new technique. So because of that, that means there's very limited number of data processing pipelines, especially for novices or people new to microED as a whole. So it's very important for us to produce some kind of pipeline that users can actually process their data easily and with minimal input. So our proposed solution to this is called AutoMicroED. So AutoMicroED starts here at file image conversion. It does indexing, scaling, merging, phasing, and final refinement to result in final structures. So what's great about our pipeline is that users no longer have to piece together different types of programs to do the full data processing pipeline. This is all in one pipeline. So we've set it up to where a user only has to start the program with a single command line call, which is shown here. So use the alias microED, and your inputs are exclusively your images from microED data collection and an arguments file. So the arguments file is really the key to the automation of our program. So the arguments file is completely optional for users, but the more information that a user puts into the arguments file, which is your metadata, so camera length, your tilt angles, your beam time, all of that goes into this metadata file. The more information that you provide, the more automated this data processing pipeline becomes. The other thing is that as this program begins, as it starts running, you're going to hit a couple pause points. So these are areas where the user will stop and check the data processing has actually proceeded correctly. If it has proceeded correctly, you simply hit enter and the program continues moving on. If this program has, or if the process has not proceeded correctly, this is a good indication, especially to new users, to look back at either their metadata that they inputted or go back to the data collection process here. So what we eventually hope to do is actually incorporate AI and machine learning at these stages to where a user themselves does not have to manually go back and do data collection changes. But if we can create a pipeline to where we can have AutoMicroED communicate with the data collection side to improve that kind of data collection. But as it stands right now, AutoMicroED is a standalone pipeline. So following this full pipeline here, you will end up with final structures, which are shown here. So these are two microED standards, carbamazepine and acetaminophen. One of the biggest advantages of microED especially is that it can handle heterogeneous samples. So this I've highlighted here, showing these red circles and blue squares. These are actually two different samples deposited on the exact same grid. So we can do data collection on the same grid with multiple samples, and all of this data can be simultaneously fed into AutoMicroED. So AutoMicroED can process multiple samples at the same time and will output separate structures out of all of the data that you gave it. We wanted to ensure that our program does in fact have the ability to handle heterogeneous samples. So with that, AutoMicroED is in fact you're able to freely access it on our GitHub page. And if you have any questions about MicroED, you can come chat with me at my poster tomorrow. And if you have any questions about accessing MicroED or AutoMicroED as a user, I'd suggest that you attend James Evans' talk on Thursday. So thank you.